undergrad at UCL, and I'm gonna do something a little different than what you've seen before. I'm gonna do a dance piece about induced pluripotent stem cells. It's not gonna be very funny, I'm afraid, but I hope it'll be entertaining, so yeah, keep an open mind. Oh, sorry about that.
At the beginning was the Word. At the beginning was Ash. At the beginning was God, was the Father. At the beginning was nature. At the beginning was Mother Nature, was Mother. At the beginning was a cell, a single cell, containing a nucleus, containing genetic information. And that cell divided, and the DNA inside of it was copied into new cells. Cells that could become any kind of cell, each of them having equal power to become a neuron, or a myocyte, or any other type of cell. They were pluripotent embryonic stem cells. But as the clones of the single cell kept dividing, they began to organize themselves in a blastula, in 3 germ layers, in tubes, and tissues, and organs. All these blastomeres contain the same DNA as the first single cell, and the only thing that distinguishes them from one another is their position in the forming embryo. As they migrate and differentiate into a particular cell type, their potency reduces until they express only a few genes to complete one function. Once this process is terminated, the fate of your cells is sealed. Or is it? New techniques have been discovered to reprogram adult cells into pluripotent stem cells. This means that a skin cell could be reprogrammed to become a cell of the cornea. This might mean that we won't need to wait in line for organ transplant when we need. This might mean that we won't need animal models to test drugs. This might mean that we won't need to use embryos to make advances in regenerative medicine. This means that the boundaries of science can be pushed further, and the fate of our cells can be newly designed. Our bodies are not static entities anymore. They are limitless.
induced pluripotent stem cells, a large population of reprogrammable somatic cells is required. Many somatic cells have been successfully reprogrammed. However, Fibroblast cells are commonly utilized because they can be easily extracted from a patient using a safe and non-invasive skin biopsy and are easy to culture in a lab. Fibroblast cells are somatic cells that produce connective tissue. Morphologically, the cells appear flat and they secrete collagen and elastic fibers that make up the extracellular matrix. Every cell type in an organism, apart from gametocytes and undifferentiated stem cells, are somatic cells. Each somatic cell has a unique structure and function due to a distinct combination of genes that the cell actively transcribes and translates into functional proteins. The difference in gene expression between different somatic cells is largely due to the structural organization of the chromosomes in each cell. Chromosomes can exist in two forms. The DNA can either be tightly wound around histone proteins to yield heterochromatin, or the DNA can be loosely unwound to form euchromatin. Loosely unwound DNA permits the binding of transcription factors and other transcriptional machinery, such as RNA polymerase, to the gene's promoter region. Once the transcriptional machinery is bound, gene transcription begins, and a molecule known as messenger RNA is made. When the DNA exists in a tightly wound configuration, the DNA is not exposed to the transcription machinery, and transcription does not occur. In this manner, the cell can regulate the genes it does and does not express. In fact, out of the 20 to 25,000 genes of the human genome, a somatic cell will only express a small fraction of the genes at any given time, and the rest of the genes are considered transcriptionally silent. This is the case for the reprogramming factors OCT4, SOX2, KLF4, and CMYC in the fibroblasts. Because these genes are not actively expressed, scientists must induce the expression of these genes artificially using transgenic strategies. There are a number of methods available for artificially overexpressing genes in a cell. The Yamenka group utilized retroviral vectors as the method to deliver the transgenes encoded for the four reprogramming factors OCT4, SOX2, KLF4, and CMYC. The retroviral vectors deliver the four transgenes into the cells. The transgenes are then integrated into the host's genome, thereby permitting its long-term gene expression. Amazingly, when the four transgenes are co-expressed in the same somatic cell, the differentiated cell is reprogrammed into a pluripotent-like state.